exercise now.
If you have growth here, these represent cells that have undergone what? Transformation. So these are transformed. Now, why do you suppose we have isolated colonies on those plates instead of lawns?
It's because they are selective.
Well, it's because the medium is selective. Did all the cells pick up DNA? Did anybody read about what's the efficiency of transformation? Even under laboratory conditions where we made our competent cells up, we're really trying to improve the efficiency of transformation. Does anybody know what % efficiency you're likely to see in the lab manual? It's 10%. Under the best of circumstances, transformation efficiency is about 10%. Now, we could have taken this exercise to an extreme degree. I could have had you do the population count, for the samples that we were using to inoculate these plates, and then you could have actually calculated the degree of dilution, how many cells there were to begin with, how many colonies did we end up on these plates, and you could actually calculate your transformation efficiency. Just take my word for it, it's around 10%. Knowing that, we wanted to make sure that we put plenty of cells on here, which is why we put 50 microliters of cell suspension on. We have enough efficiency that we get to see some colonies. But there's another little interesting feature of these colonies. Hopefully you'll be able to see this. Most of the ones that I looked at showed at least some evidence of this. Here will be a colony, and around the outside, there are some little bitty colonies. So these are transformed. But these guys outside, these are called feeder colonies. Or they're called satellite. They are not resistant. So what's going on out there? Well, let me give you a hint. The medium that we have contains the antibiotic ampicillin. And our plasmids are carrying a gene, the marker gene. This marker gene encodes beta-lactamase. It's actually called the BLA gene. So the marker gene is the BLA gene, and BLA stands for beta-lactamase. What do you suppose beta-lactamase does? It breaks down beta-lactam antibiotics. And ampicillin is one, so it breaks down ampicillin. What's happening is these transformed cells are producing beta-lactamase. And again, pay attention to enzyme names. Oftentimes the enzyme name tells you what the enzyme does. So beta-lactamase breaks down beta-lactam antibiotics. In this case, it's breaking down ampicillin. The colonies here, this colony is producing this substance, and it's going into the medium. What does that tell us about beta-lactamase? Is it an endoenzyme or an exoenzyme? exoenzyme, they're dumping it into the environment, which is then going to allow the little feeder colonies to show up. This would only be significant if you were working in a laboratory situation where you wanted to make sure that you were actually getting the colony that was transformed, and not all these little guys around the outside that are not. So beta-lactamase is an exoenzyme, and they're dumping it into the environment, it's diffusing out. You'll get more and more of these colonies if these guys were allowed to incubate for a longer period of time. So it's going to be a fairly short period of time that they get to grow here. Well, okay, so we were successful with transformation if there were colonies on these plates. Some of you that were in here kind of as the exam was coming to a close, I had pulled those plates out of the incubator and was looking at them. That's because you get points for transformation. If your plasmids did not show up in the gels, but your plasmids show up on these plates, and one colony will do you. If you've got one colony on a plate that has epsilon, that means you had at least one cell that was transformed. Now, here's the question. If you're a group of 3 people and there are only 2 plasmids, how do you want that graded? Did you actually do the preps individually? In which case, do the people who did the preps have their names on them? Or do I divide the prep, calculate mathematically how many points you get? If your group has only 2 people in it, and you got 2 plasmids, you're good. Because each person gets 5 points. If your group is three people and you only got 2 plasmids, either somebody doesn't get a full 5 points, or we can divide it up.
As colonies on the plate, right?
Yep.
So
Colonies on plates.
what if all of your plates have colonies?
If all of your plates, well, you answer that question. If you've got this, what does that mean?
That's 5 points for everyone.
That's exactly right. If your 3 GSAM plates, in other words, you have colonies on the PGEM plate and the PGLO plate and the QC9 plate, then all of your plasmids were there and everybody gets 5 points. It's the groups where there's missing 1 plasmid. It's usually PUC19, which is surprising because that plasmid is the smallest of these plasmids and usually it's picked up very readily. But again, it all had to do with the extraction procedure and whether you were using the pipette correctly and putting the chemicals in correctly and all that.
Maybe we just stop with like 1 point. Give everyone 4.
No. It's gotta be more accurate than that. Well, I'll have to think about this since you guys can't give me an answer. Now, there's more to this exercise because there was another plate. But are you all following me so far and what this data shows us? And whether you were successful with your transformation or not. So supposedly about 10% would be a bunch of colonies. If you have only 1, I'd say your transformation efficiency is significantly less than 10%. But hey, it worked. If it's 1%, it doesn't matter. It's there. So that's all you need. Now, if you look on page 229, you see a little diagram that says figure 22B.1, operon controlling rhabinose utilization. So we have a regulatory gene over here. And all these little boxes are actually pieces of DNA. It looks like a train, but not. Okay, so this looks like a set of train cars, maybe, but if I draw it like this, this is DNA. So a segment of DNA that includes regulatory sections, such as a promoter and an operator, and a series of structural genes. What is this thing? It's an operon. It's not just any operon, it's the arabinose utilization operon. this operon works a lot like the lactose utilization operon, except that it's not quite so complex. So this regulatory gene, which is called ARIC, encodes a repressor. It's constitutive, and it's active alone. If the repressor is constitutive, that means it's always being made. If it's active alone, what does the repressor do? Binds to the operator site and blocks transcription. And it's going to be there because it's always being produced, so that repressor protein is going to be sitting here, blocking transcription. So is this an inducible operon or a repressible operon?
Inducible.
It's one that's off, but can be turned on. It's inducible. So this is an inducible operon. Given what our last plate was, what do you suppose the inducer is?
Arabinose.
Arabinose. This is a little bit different than the lactose utilization operon because lactose wasn't actually the inducer. Lactose had to be acted upon by an enzyme and converted into allolactose. Apparently not so much. With arabinose, it's straightforward. It's the inducer. The function of the inducer is to bind with the repressor protein and inactivate it. Here you guys probably thought that that was done with exam 2 and that you didn't have to worry about it, but no, it's going to come back on the laboratory exam 2. We're going to have operons we have to deal with. It's a little bit different because you don't have to have a modified sugar. It's just arabinose. It's the inducer. But the inducer is going to bind to the repressor and inactivate it. Then the repressor falls off the operator, and we're going to be able to express these structural genes. Well, it turns out that in this operon, there's been a gene added. It's the gene for green fluorescent protein. This was added to that operon. It turns out that the gene for green fluorescent protein comes from a jellyfish. Is a jellyfish eukaryotic or prokaryotic? This is a really hard question. Eukaryotic. Now, this next little bit of conversation is really going to tax your thinking ability. If I want to take the gene from a jellyfish and stick it into E. coli, can I do that? Can I just go to the jellyfish and take jellyfish DNA? Yeah. First of all, if this jellyfish gene is in a jellyfish, it's transcribed, right? And then what? What happens to messenger RNA in eukaryotic cells? Is it? I mean, it is eventually, but is it as a transcript product? No, because eukaryotic cells have spliceosomes and they do post-transcriptional modification. So this messenger RNA is modified which means introns are taken out. And I did use that word, did I not? Spliced together, that was your hint. Lysosomes do this. Small RNA or small nuclear RNA, ribozyme. Well, anyway, bacteria can't do this. So how are we going to modify our jellyfish gene? Turns out what we have to do is get the messenger RNA after this process. Get your functional mRNA. We can go into a jellyfish and find functional messenger RNA. We can capture it because the other thing that happens to the messenger RNA is it gets polyadenine tail. And that makes it very recognizable. And so you can make a column with a bunch of poly T's attached to glass beads, run jellyfish extract through there, and what will get trapped on there are RNA molecules. Presumably they have to get it from a specific part of the jellyfish to get the right one. But they're wanting to get that specific functional messenger RNA. And then what do you do with it? Because we want a gene, which is DNA. Reverse transcriptase, virus enzyme. So we subject it to reverse transcriptase and we get DNA. And now we know that reverse transcriptase doesn't just copy the messenger RNA into one strand of DNA, it then degrades the RNA and replicates that DNA and makes it a double-stranded molecule. So now we have double-stranded DNA. It's the right gene, but can we put that into E. coli? I mean, we can put it into a cloning vector. We know that we have to cut the cloning vector with the right restriction enzyme, and we have to cut the borders on this piece of DNA with the right restriction enzyme. But is there something else that's going to get in the way? Remember I said bacteria have restriction modification systems? So they have restriction enzymes. The function of restriction enzymes is to chop up foreign DNA. They're primarily a defense against viruses. Viral DNA is recognized as foreign. Their job is to chop it all to pieces. What do the modification enzymes do? It's like, oh man, that was lab stuff back there a while. Modification enzymes modify the cell's DNA so it cannot be cut. I'm not sure this material is in the lab manual. So is it significant? Will it possibly show up on laboratory exam too? Oh, yeah. So modification systems are modification enzymes. They modify the cell's DNA. They're methylases. So they add methyl groups to the recognition site. Well, so what this means is that this DNA that came out of the jellyfish then has to be modified. You're going to have to subject it to these methylase enzymes, make sure that it won't get chopped up, and now we want to put it into our plasmid. By the way, the plasmid we want to put it into is PGEM. So we're going to cut PGEM. permanently closed circle, and you cut it once with a restriction enzyme, it's gonna open. And you're gonna linearize it. And now, as long as we have complementary cohesive termini, so we have this little piece of DNA modified so it's got cohesive termini out here that will match the plasmid that we've now cut open. Now we can stick that in there and it will hydrogen bond in place. And if we want it to stay put, what enzyme do we use? Ligase. I'll put it in there, make it green. Keep in mind that green fluorescent protein gene was stuck in to the erasmus operon someplace in the vicinity of those structural genes. It had to be behind the promoter. It's also behind the operator. So we had to use ligase to stick it in there. Now it's in there. Now we can use that cloning vector and put it into the E. coli, and your E. coli should be expressing green fluorescent protein. Are they? Where would we find that out? On that plate. Does anybody know about fluorescent molecules?
No. There.
If you want to see fluorescent molecules, what do you have to expose them to? Black light. They're fluorescent. They'll only show what UV light. So now I'm going to impose upon you guys and ask you to come into the back room one table at a time. When you come into the back room, what I want you to bring is your CSA amparabinos plate and your you could bring your plate that has DH5 alpha and P-glow, but no arabinose. So we can compare the two plates.
DH5.
So table one, it's going to come back here.
DH5.
Yeah. Table one.
Yeah, she said table one, so let's go. But which two plates are we bringing?
The one with the enzyme, and then the other one with the P-glow.
Yep.
CSA with P-glow.
TSA with P-glow. So that's P-cam. Is this P-glow? No, that's P-glow. This one. Okay.
Oh, and then your own console.
Table two, some left over, so I didn't know who got them and who didn't. Does anybody know? You guys remember where you got
Yes.
if you got those pictures or not? Okay, so nobody knows they didn't. Okay.
I got mine.
Okay,
If you feel left out right now, speak up.
unless you're not here, in which case you're definitely left out.
Stay right here.
Yeah, these were passed out on Tuesday. Yeah. Okay, so now we're ready to do the virus exercise. Now that you've been thoroughly stressed out by transformation. All of that is fair game for questions on Lab Exam 2. Probably won't all be on the quiz on Thursday next week because I can only answer the questions as part of Company Viruses. So now we're going to do virus exercise. So, probably the most important thing to remember about the viruses we're working with, they are bacteriophage. And what are bacteriophage?
Viruses.
Right, they're viruses that infect bacteria. They cannot infect you.
Virus finding.
That's amazing.
One of the things that I told you about bacteriophages, they tend to be very
You're not
host
touching.
specific.
Oh, you're trying to move it. Just take it.
I know, I was trying to think what to do.
Just leave me alone.
There was a question on the exam that talked about why are they host specific? What's the stage where this comes into play? Absorption or attachment, and what makes them host specific? The receptors. The receptors on the surface of the virus that have to interact with specific receptors on the surface of the host. Consequently, bacteriophage can be used for phage typing. You bring up the tax because we talked about bacteriophage typing way back when I was talking about different criteria that could be used for classification. But it's basically the process of using known types of phage to identify unknown types of bacteria. For this particular exercise, we're not using just any bacteriophage. We are using colophage. What is a colophage? So the coli part of this tells you this is a virus that infects E. coli. And this is significant because in order to see the effects of bacteriophage, you have to give them a host cell type to infect. Because bacteriophage are submicroscopic, you cannot see them with our light microscopes. All we can see is evidence of where they have caused damage to the cells. We're going to be using two colophages that are cytolytic. And what do we know about cytolytic phage? They go through that cycle that ends up with the host cell releasing them. The host cell is lysed. Cytolytic means cell lysing. So they're going to kill their host. They're going to cause the formation of structures on the surface of the plate called plaques. And I'll pass these out. These are going to be used for the second exercise, but you can pass them now so that you can look at them. Don't get too excited about the label on these plates. These plates are labeled simulated sewage.
Is she thinking calm?
Just to let you know it's not simulated sewage. So the clear areas that you see on the surface of these plates are plaques. And plaques indicate regions where the bacteria are killed by the phage presence. They kind of look like little windows. However, if you look at the plate from the front, from the top side, what you'll see is they're more than just clear spaces. They're actually divots. They're like little indentations. So cytolytic phage will kill their host, and they cause the formation of plaques. So those plaques are regions where the cells are dead, and as I said, they're actually divots. The lawn, which you see around that, is the bacteria. But these plates are made by a method that you guys haven't done yet. So we've done streak plates, and you've done plates where you put liquid on the plate and then spread it around with a glass spreader. Done that a couple of times. Tonight we're going to be doing pour plates. And what pour plates involve is using a substance called top auger, and you have to put into that your bacteriophage and E. coli, and then you pour that on the plate. So we're going to be using some media. The plates are called bottom auger plates, and you're going to be putting phage and E. coli into top auger, and then basically you put the top auger onto the surface of the bottom auger, and that's how you get this effect that you can see on those plates. So you're going to be making plates somewhat similar to that. But before we get started, because we have to have the top auger has to be liquefied, and right now the top auger is solid. For this exercise, for this set of exercises, you need 7 tubes of top auger melted. My recommendation would be that you melt more than 7, because if somewhere in the exercise you mess one of them up, you don't want to have to go back to the very beginning and start over again. So my recommendation would be to put 9 top auger tubes in your beaker, and I'll do a demo here. So 9 top auger tubes will fit in here just nicely. And then you want to put water in the beaker that does not come up above the level of the auger. These tubes contain 3 mils of auger, so the water level should be right equal with the level of the top auger in the tubes, so that when this is heated, because you have to boil it in order to melt the top auger, and you don't want to let that water get up to the caps, where it can then, by capillary action, go right up inside the tube. Because you want your tubes to stay sterile. So make sure the water level in your beaker is the same level as the top auger. Okay, so each group, and again, you're going to be working in groups like you did with the transformation exercise. Each group needs 9 top auger tubes in a beaker with water. Put them on the
Grab the one
burners.
next
Put
to
them
Sunny.
above the
No, grab the
burners
one
on your
from
racks
Sunny.
and get
Grab
them
Sunny's
moist.
stand.
Sunny
I wasn't
was
gonna move
the
the stand, I was gonna pause.
Oh, I didn't.
4 bottom auger plates per group, and the bottom auger plates are currently in the incubator because it helps if they're warm for this exercise.
You take it on first at like a certain time.
There's like a zillion dates.
Oh,
For
it's not like yours.
But there's only like
The ones that are, I'm gonna put these over here on the side bench. The ones that are not in a
Yeah, I would
bag,
do it over summer.
Sasha left in the incubator for
You
us,
wanna be able to sit
and
and
they're
study for
fine.
them?
Yeah.
They're clean.
Yeah. Yeah, don't take it before you take the exam.
And because I'm taking so like that's why I'm staying extra semester too.
So each group needs 4 bottom auger plates.
We'll take open.
yeah.
Which game did you guys take?
Have you not taken it yet?
No, I took it.
What camera are you in, Sunny?
Did you drop out? Did you turn on
What
the
camera are you in?
finish?
Oh, 3D, that's right.
Or you guys did?
Literally still just like.
Okay.
And you feel like you did good? Wow, I hate you.
Well, I mean, I was unsure. I was like, I had to.
Yep.
But
Five?
last night I was like,
No, my grade's not better when yours.
what's your yesterday?
My grade is way better, but yours being worse.
I failed too though, honestly.
I know you were sad.
Huh?
I said you were sad. Extra.
No, I don't. No, it wasn't me. That was the guy next to me. Chick behind me.
This one's my
I wasn't even.
Or when she was like, she had a pencil and she's like,
What do we do?
Talk to the lab leader.
Okay, can I have your attention? You're waiting for your top auger to melt. So what's gonna happen is the water in the beaker's gonna start to boil and the tubes are gonna start jumping around in there. If you've ever boiled things in chemistry where you put the boiling beads in there, these tubes are gonna act like boiling beads, so they're gonna bounce around. Don't worry about it. But you wanna make sure that your top auger is completely liquefied, because if it isn't, when you try to do your pour plate, you're gonna get chunks, and that's not okay. So you want these to boil long enough that you know that top auger is completely melted. And once you have done that, I want you to transfer your top auger into the water bath, which may require that you make a paper towel pot holder to move your beaker to the water bath.
What?
The water bath is over here.
You want us to move the entire glass?
You're gonna move the beaker to the water bath. I mean, the alternative is to bring the tubes over one at a time,
No.
but they're hot little suckers.
They're exploding.
You're gonna put them in these racks over here. If you want to label them, you can. Don't write on the plastic. You'd have to use tape. Now, the bottom of the plates that you're supposed to pick up, you're supposed to label, see page 236. It helps to label both sides, but it's not terrible for.
Okay, we need to label these. You want me to label it? Okay. Turn to 226 in your
And
lab.
while
I told you I
you're
don't
doing
have
that, I will set up your virus activity kit.
Sunny, you're just bubbling like one big bubble at a time.
Oh.
What page?
You found me for.
Oh, 236.
Scrolling over here.
Okay, and then, so do I need to take these over right now? Or when they start jumping more?
No idea. Talk to the
How
lab
much
leader.
jumping should they be
ours is
doing? Mine's like, ours are jumping about the same. So do we go over now?
Whoa. What really jump? Good jump.
How much jumping do they need to be doing?
Or,
What
are you
auger?
good doing it? Yeah. Okay.
What auger is this?
Yeah, you can take your time. It's not like it's a race or anything.
Okay.
But yeah, unless you can coax somebody to
The
join you
race
from
of
another group.
life.
What kind of?
Borrow a lab partner.
What kind of auger do I say?
It's bottom auger.
Just bottom, that's it?
That's the bottom.
Oh.
Bottom auger.
Bottom auger.
Can I have your attention up front?
Mm-hmm.
So here's what we're going to be doing next. You have your 4 plates, your bottom auger plates, and they're labeled. This isn't a good thing because the plates tell you exactly what's going to go on there.
What?
To inoculate a plate, you always do this. You remove 1 tube of top auger from the water bath. You bring it to your workbench. You add 200 microliters of E. coli as indicated by the label on the plate. It's either E. coli number 1 or E. coli number 2. But if you're doing 200 microliters, what pipette are you using?
So, yellow, white,
Oh my
blue,
goodness.
red,
Blue.
pink.
Turquoise. Blue.
Knew I'd get there eventually.
Remember, set it to 200 microliters. Okay. Then you're going to add 100 microliters of colophage. That's yellow.
know, why is we should get that.
And then you're gonna roll the tube between the palms of your hands. Keep the tube upright, put it between your palms, roll it, roll. And then you're gonna pour it over the surface of your bottom auger, the plate that is properly identified based on what you just put in that top auger. And you're gonna gently tip to make sure that the bottom auger covers the top auger covers the entire surface. Once it has covered, leave it alone. Don't keep messing with it. If you've ever tried to stir Jell-O that was partially set, you'll know what would happen to auger if you keep messing with it and it cools. It'll just make a rumpled up mess. So once you've covered the entire surface, then leave that plate alone. Okay. Now, do you need additional instructions?
As of now, no.
No, because your plates are labeled. Your plates tell you exactly what goes on there. You get one tube of top auger out. You add 200 microliters of E. coli. You add 200 or 100 microliters of paste. Roll to mix, pour on the bottom. Should you have your Bunsen burners going?
No.
Yes, because it was all happened to be done aseptically. You do not want to introduce a bunch of things that would grow on there other than E. coli. So yes, aseptic testing matters.
I don't think you know. I didn't write
I think I told you guys. Unless it wasn't gonna be me.
The thing, if it's around.
So yes, have your burners going. Your plates are labeled, they tell you exactly what goes on there. You guys should be able to do this.
I do have a question.
Okay? There's an E. coli seed in your virus activity kit. It is not part of this exercise.
There we go.
Leave the E. coli seed alone. You're using E. coli 1, E. coli 2, and the phage, which are in the little kit and they're properly labeled.
Somebody just keep taking my tablet to it.
You guys should have blue pipette, yellow pipette.
Are you stealing my wood too?
Yeah, too.
Oh, you trying to steal mine now?
Okay, do this, and then we can do this.
Oh, I don't think we're streaking. Yeah, we're not streaking. We're doing the. I bet that's the same citation, guys.
Oh, okay. I'm like, where are the other 5 photos? I got you.
But we had it extra.
Alright, Gabe.
Make sure you set this
Your
right.
job is to be the lab leader. No, Okay.
I have 3 of those now.
Okay, E. coli indicated by points.
Wait, what are we doing?
Okay, E. coli 1, E. coli 2, E. coli 1, E. coli 2.
I'm so lazy. I'm so
And,
Go watch her not tell them.
I'll
That's your
give you
job.
your first listing. Oh, after I look at it, but I have one plate labeled 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. And you need one plate of simulated sewage, which you already have. And your top auger is already melted and it's in the water bath. So you may get these things.
It's so stressful.
Seriously, this is exactly wishing this fucking food blogger.
So what are we doing? Grabbing.
Just 4.
No. Yeah, don't worry about it.
To my eyebrows.
I mean, you can kind of pay attention to what it looks like so that when you're counting blacks, you know that. But actually, for the fish type, you don't count them anyway. So it doesn't matter.
I see on the next step.
Yeah, but I didn't say that because if you turn them over too quickly, you just mess them up. You wanna make sure they stay there until they're set. If you keep messing with them like that,
You're like increasing mine in
you know, that's not,
Light voltage.
that started to set up before you got it. That was fine. 4 tubes of KCl broth, they're 4.5 mils, over here. You need 1 tube that has 1 mil. The tubes that have only 1 mil of KCl broth look like this. They're very different. Little bitty tube. You need 3 bottom auger plates. You're gonna label those 3 bottom auger plates 10 to the minus 3, 10 to the minus 4, and 10 to the minus 5. And you need 1 plate of simulated sewage. The simulated sewage was passed out to the leach treatment house. Now, what you're going to do with this, to start with, you're going to use your wire needle. We've never used the needles before. We always use loops. So for this exercise, you want to make sure you're using the straight wire. And what you want to do is stab a plaque on your simulated sewage plate. So these represent the plaques on here. Try to make your stab straight down. Don't come in at an angle because it's going to change the number of virions you pick up. So you want to come straight down and out. After you've stabbed 1 time, rinse the colophage off into the 1 mil of KCl broth. I tried to tell people to shake it off, but then what they were doing is putting the needle above the liquid and shaking it back and forth. That doesn't work. You have to actually put it in the liquid, rinse it off. Then go back and stab it again. I want you to stab 3 to 4 times. Make sure that you record the number of stabs. Don't stab more than 4. Each time you stab, rinse the colophage off into the KCl broth. After you have done that, you're going to set up a serial dilution.
Can you use equal 1 for open?
We did a serial dilution before, so this is not terribly difficult. You know that a serial dilution involves transferring a specified quantity of liquid from one container into another container. And the container you put it in has to have a specified quantity. So we're going to start out, this is what we're going to dilute. When you dilute bacterial cultures, you could have done the serial dilution, which we did for our population count, or you can streak a plate. I don't think you can streak a plate with colophage. So instead, you use this dilution procedure. You want to set up a serial dilution. This is what you're diluting, your 1 mil of KCl broth. Your serial dilution is going to be a series of 4 tubes that contain 4.5 mils of KCl broth. They're supposed to all contain the same amount. Okay, so this one is going to be 10 to the minus 1. This is 10 to the minus 2. This is 10 to the minus 3. This is 10 to the minus 4. Each one of these is 4.5 mils.
Just 10 to the minus 1.
And what you're transferring into it is 500 microliters. So KCl broth, you're going to pick up 500 microliters of it. Using a blue pipette, you're going to transfer into here. This is from the KCl broth. If you put 500, which is 0.5 ml, 500 microliters is 0.5 ml. If you put 0.5 into 4.5, what do you get? 5. Therefore, a 0.5 into 5 is what dilution? 10 to the minus 1. 110. 10 to the minus 1. Therefore, 500 into 4.5, that's why that tube is labeled 10 to the minus 1, because it is. Once you've put that in there, roll to mix. Roll it between the palms of your hand. Remember, keep those guys upright. The snap caps leak. After you've done that, pick off that pipette tip, get a new pipette tip, transfer 500 microliters from the 10 to the minus 1 tube in the tube labeled 10 to the minus 2. Make sure you change tips. Put that in there, again, it's the same dilution, it's 10 to the minus 1. Roll to mix. After you've done that, you get a new tip, take 500 out of here, put it into the next tube. Roll to mix. And after you've done that, take 500 out of here and put it into the next tube. So you're going to end up with these dilution steps. The next thing that you're going to do is take samples from the last 3 tubes and put them on plates. So this is going to get on a plate, and that plate's going to be labeled 10 to the minus 5. And this one's going to get on a plate, and the plate is going to be labeled 10 to the minus 4. And this one's going to get on a plate. Yeah, I've got my numbers on the wrong side, but that's okay. This one's going to get on a plate, and the plate is labeled 10 to the minus 3. So we're only plating the 10 to the minus 2, 10 to the minus 3, 10 to the minus 4. They're going on the plates. The plates are labeled 10 to the minus 5, 4, 3. The reason for that is because to inoculate the plates, you have to do this. To inoculate a plate, remove 1 tube of top auger from the water bath, add 200 microliters of E. coli seed. And then you're going to add 100 microliters of coliphage as indicated by the plate label, except that the plate label doesn't match the tube label. Just remember, because we're adding 100 microliters is 1/10 of a mil. So the volume that's coming out of the tube and going onto the plate is always 1/10. That adds a 10 to the minus 1. So this is a 10 to the minus 4 solution. You're only taking 1/10 of it, which makes this a 10 to the minus 5 plate. Make sense? So the first thing you have to do is set up your serial dilution. First thing you have to do is stab your plaques. Record your number of stabs, then set up the serial dilution. Then make sure your plates are labeled 10 to the minus 3, 4, 5, as shown on page 243. And then just remember that you always have to inoculate your plates like this. Don't deviate from this. Remove 1 tube of top auger, 200 microliters of E. coli seed, 100 microliters of phage. Roll the mix, pour over the surface.
So we're transferring between the two, we use the blue pipette and 500 microliters to use.
Yes, so you're going to use the blue pipette for these 500 microliter transfers. Make sure you change the tip after each step.
And then the 1 milliliter of the original, do we take 500 microliters for this as well?
Yeah, that should be 1 mil in there.
Yeah.
I hope it is.
I
hope it is. That's what it says it is. It looks kind of scrawny to me, but no, they should be
Will you
1.
grab 2?
We have to boil auger.
It isn't.
Why?
So we have extra.
Yeah.
Gee whiz.
Oh.
Double check your little yellow tube. I just picked up a yellow tube up here that has 500 microliters on it. Your yellow tube should have 1 mil. That means it should be 3/4 full.
Where's ours? 3/4.
Somebody takes their phage and squirts it on the plate. Don't do that. Your phage goes in the top auger. From your dilutions here, these dilutions, even though I drew the arrow pointing to the plates, they're going to end up on the plates, yes, but you do not squirt them on the plates. You put them in the top auger. Always inoculate your plates.
Sorry, we don't have enough. We have to boil more auger.
Two top augers.
Yes.
Yeah, we need to boil 2 more
So
top
it's
augers.
not
Can you do
500,
that?
it's 100.
before everyone takes it all.
Yeah, your dilution.
Gotcha, alright.
Okay, so I wanna might be thinking a bit too comfortable. So I have one part stop 4 times. So wait a second. So when we stop every 3 minutes. Okay.
Jab it in.
And shower again.
I don't want to burn that.
Okay.
Oh, it's fine. You can put it over. It's not gonna burn it.
Okay. And also as for
They burned that.
a
Even hold for this too.
Oh, everybody did mine.
okay. So
But if yours was chunky like that, was it because you let it sit around too long?
I think
Or was it not thoroughly